Hello, thank you everyone for joining us today. We really appreciate you taking time out of your schedules, and I know the holidays are fast approaching, so we appreciate you taking time to be with us. Microbial Insights is really delighted to have Dr. John Wilson with Scissortail Environmental, who's going to be talking for us today. And I am always personally delighted to have the honor to introduce Dr. Wilson. I've known him for many years, and to be honest, he needs no introduction, as most people in the field, including myself, either talk to him on a regular basis for guidance or use one of the many documents that he's written. He has a spectacular history from 1978 until recently. He worked as a highly respected research microbiologist at the R.S. Kerr Environmental Research Center in Ada, Oklahoma, where he led research dealing with biological and non-biological processes that destroy contaminants in groundwater. And then after his retirement, Dr. Wilson continues to work. He founded Scissortail Environmental Solutions. And today he's going to be talking to us about a topic that I think most of you will be really excited to hear. He's going to explain how molecular biological tools can be used to predict rate constants for anaerobic biodegradation of chlorinated compounds. So I can't wait to hear this talk. Please note that, as you can see, the webinar is being recorded and it's going to be available in the resource section of the Microbial Insights website within a few days. A link will be emailed to everyone who registered for the webinar, so you don't have to contact us for that. You'll get that automatically tomorrow. And then after the presentation this morning, you're going to have a real opportunity because we're going to open up a short question and answer session. So if you have questions for Dr. Wilson as he's talking, please enter them into your question section of the GoToWebinar panel, and we'll turn it over to John and get him to answer those. So without any further ado, I'm going to go ahead and turn it over to you, Dr. Wilson.
Well, Dora, thank you for the opportunity to address this group. Let's go to the next slide. What I'm talking about today actually came out of the research product of two ESTCP projects. The first one was led by Dr. Mandy Michelson with the Corps of Engineers, an effort working with Kate Kasharik at Battelle, Paul Hatzinger at Aptum, and Frank Loeffler at University of Tennessee to actually evaluate and validate an advanced molecular biological tool that involves measuring peptides for active proteins. Next slide. And I'll also talk about a computer application that was developed under a project that Tony Danko with NAVFAC led. The technical principals were Dave Adamson and Charles Newell at GSI Environmental. So just wanted to acknowledge all the people that went into the effort of producing the information I'll provide today. All I did is pull it together and put some words and slides around it. Next slide. I need to explain to you why you need to care about rate constants first. Well, maybe I don't, but I'll explain how you can explain to your decision officials and your bosses and your regulators why they need to care about rate constants. The US EPA approach toward accepting natural attenuation as a remedy is described in the 1999 OSWR document described here. And it's based around the idea of lines of evidence, and it usually requires at least two and sometimes a third line of evidence to establish and document that M&A is an appropriate remedy at a site. Next slide. And the first line of evidence is basically a clear monitoring record that it's going away. And so it's based on the empirical observation that concentrations or quantities are declining over time. Next slide. The second line of evidence basically rationalizes and explains the first, that, well, we would really expect that to happen. And so it's other kinds of evidence, often indirect evidence, which demonstrates the type of processes that are acting and the rate at which the processes will reduce the contaminant concentrations to the required levels. That rate is required because the EPA needs to know not only is it happening, but is it happening fast enough to do any good and get us out of this site and get it closed. Next slide, please. So this is where we are, and this is where I'd like to see us go. Right now, most of the time, people use the qPCR tools to interrogate the environment and see if there are organisms there that have the capacity to degrade cis-DCE and vinyl chloride in anaerobic groundwater. I say basically this is anybody home question. But what we don't do is take it to the next step and see if the abundances of these organisms would explain the rates. Another way of saying that, not only are they at home, but are they doing enough good to help us get out of this site? So how much activity are they exerting? So our current state of practice provides only the first half of the second line of evidence. Let's go to the next slide. And so basically that just kind of illustrates that. The bold black is what we are doing, and the red font is what we really should be trying to do with our qPCR data. Next slide. Made an initial stab at this in an earlier ESTC project that was led by Carmen LeBron. It's just a component of a sort of an expert system to try to incorporate various tools that allowed you to make a quantitative estimate of the rate constants. Let's ask the question, did the abundance of the biomarkers provide a plausible explanation for the field scale rate constant that would apply to allow M&A to be an effective remedy at a particular site? Next slide. And the approach we took was entirely empirical. We simply calculated the first order rate constants for attenuation with distance along the flow path at the site of concern and plotted that against the abundance or the density of the halococcoides cells recognized by the qPCR marker for the ribosome. And so that blue shape I offered as well, if it falls inside the blue shape, then you can take that as the second line of evidence that the rate constant explains the field scale behavior. Next slide. And it didn't work. Most people that I've dealt with, particularly regulators, do not find this satisfying. So the empirical approach has not gained a widespread application. And so we're going to try again using a deterministic approach to see if it's a little more credible. Next slide. So we're going to go to the familiar Michaelis-Menten equation, basically a hyperbolic function that compares the rate constant to the quantity of active agent, the concentration of compound of interest, and a half saturation constant that describes the hyperbolic function. There's a lot of this information in the literature for a lot of different systems, and it is traditionally normalized to the amount of protein in the active organisms. Well, that's useful if you've got a pure culture in a laboratory, but we really don't know how much protein, can't measure how much protein directly right now easily is associated with all of the halococcoides cells in an aquifer sample. So we need to modify this approach slightly. Next slide. So instead of having Vmax be normalized to protein, we need to normalize it to the number of gene copies or to the number of enzyme peptides in the environmental system. Next slide. There are actually 3 different tribes of halococcoides organisms that we find in environmental samples, and they're called clusters. They're loosely named for the place of the original isolation of the first example of each cluster. Pinellas, Florida, Victoria, Texas, or Cornell is Ithaca, New York. And these 3 cultures actually were were very typical of a number of other strains that show up as you analyze dihalococoides strains from different sites across the country. Next slide. The three different clusters have some important differences in terms of the enzyme types that they have. And there was sort of a confusing or confounding little myth that came about in some of the early enzyme studies. We used to think that if the cell only had the TCA reductase, that one particular enzyme, it couldn't grow up using vinyl chloride. And that if it grew by the degradation of vinyl chloride, it had to have two other genes, either the VCRA gene or the BVCA gene. And the importance of that is we thought that TCA gene just didn't simply do cis to vinyl chloride, and so we'd have it stall at the level of cis. Well, this is what we used to think. Next slide. There's two recent papers that have taken another look at this, and what they've basically discovered, it was a vitamin deficiency in the culture studies. And if you gave the cells enough cobalamin, they do just fine using the TCA gene to grow on vinyl chloride. And so we now know that this TCA calls a cis stall business is probably not at all true in environmental procedures and environmental behavior. Next slide. But between the Victoria and the Cornell strain, the Victoria strain has the VCRA gene that really does do a good job on cis to vinyl chloride. And the Cornell strain has the TCA gene that we now know makes an enzyme that can do vinyl chloride, but it doesn't have the VCRA gene. But they're two different bugs or two different creatures and how they process cis-DCE and vinyl chloride. Next slide. It turns out that there's some differences in the values for Vmax and Km for cis-DCE and vinyl chloride. And so what I did is basically for the rest of the talk, I'm going to work it up two different ways, using the kinetic parameters that are appropriate for the Ithaca strain, the Cornell strain, or the kinetic parameters that are appropriate for the Victoria strain. And I gave you the numbers that I'm using and the primary reference where I got the values for the parameters. Next slide. And so what we're going to do is come up with a Vmax that's normalized to the gene copies. And then we're going to multiply that by the abundance of the gene copies to calculate V, the rate constant for the reaction. Next slide. I'm going to kind of go off in the weeds a little bit. The amount of TCE that's in the water is not all the TCE that the bugs have to deal with. And we have to come up with a correction factor on the amount of TCE that's available ultimately for degradation, but is hiding at any particular point in time in the sorb phase. And so we're going to use the retardation coefficient, R, as conveniently used to describe flow in groundwater to help us figure that out. And I calculated R from the fraction of organic carbon, the partition coefficient, the bulk density of the solids, and the fraction of pore volume that's occupied by groundwater, as is traditionally done by the geohydrologist. Next slide. So I calculated an equivalent concentration, which is just simply the measured concentration S multiplied by R. And so E is the amount of TCE or cis-DCE or vinyl chloride that the bacteria have to actually deal with, as opposed to just S. Next slide. So we're going to calculate an apparent first order rate constant, apparent pseudo first order rate constant, by dividing the rate of degradation by the amount that's available to be degraded. And that amount is E, the equivalent concentration. And we end up then with a K in units of per year. Next slide. So what we did is attempted to validate, evaluate, benchmark all of this against a real field site. This particular site is probably one of the best studied, best known sites that I've seen anywhere in the literature. It's a relatively simple site, and the monitoring wells transverse almost all of the flow in the aquifer, and they're reasonably spaced along the flow gradient. Next slide. And there are a bunch of wells. So this site is densely characterized, relatively small, the geohydrology is simple, and high density of data points. Next slide. The other advantage of it is it's been studied for a long time. And so the flow velocity of this site is relatively fast. It'll sweep out the area sampled by the monitoring wells in somewhere between 1 and 2 years. And so when we go to 2007 to 2013 to 2016 to this year, we're looking at several flushings, if you want to call it that. In other words, the poor water's been replaced several times in this period of time. All right, so we're going to use various wells. Different wells were sampled in different years. And we're going to calculate first order rate constants for disappearance of cis-DCE and vinyl chloride in this plume at field scale. Next slide. This is the figure in the upper right is for cis-DCE for 2007 and 2013. The one in the lower right is for 2016 and 2021. You can just look and see that now the fit is not perfect, but there's a reasonable correspondence between a fitted line and the measured data points. Then the lower left is data for vinyl chloride in the 2 years that those data were available to be calculated. And then the table in the upper left then just simply summarizes rate constants that are fit to the data with an 80% confidence interval on the rate. If you come back and take a look at this, you can make your own judgments about how close that is. But these are actually about as good as anybody ever gets at a field scale system. Next slide. All right, so the first thing to learn from this is that the rate constants are reasonably consistent over this 14-year interval. It's not changing a lot. If it is changing, it's going up maybe, but really not a whole lot of evidence that it's changing at all. And that the rate constants for cis and vinyl chloride, particularly within the limits of confidence, are not different. Next slide. All right, so we've got a system where the biological reductive dechlorination processes are consistent over time and space. Let's see then if we can predict those rate constants from the abundance of their qPCR markers. And so the system was sampled in 2013, and these are the data for the DHC, for the ribosome marker, for TCA, BVC, and VCRA. The first thing to notice that these numbers are actually pretty high, sort of at the high end of anybody's experience. There's a lot of dihalococoides in this system. The other thing to notice that it's not the same number all the way down the flow path. It changes. Next slide. So we use the kinetic parameters, the concentrations of the contaminants, the abundance of the biomarker, and a retardation coefficient to calculate a rate constant for biological reductive chlorination in the groundwater that was sampled by each individual monitoring well. And so that's in the next slide. And so the upper slide is the data that was used to calculate the rate constants. And I'm sorry, the upper table is the data used to calculate the rate constants. And the lower table are rate constants that are extracted using the kinetic parameters for the Victoria strain or the Ithaca strain 195. And basically those use the equations and the parameters that I provided to you earlier. Next slide. And so what do we learn from this? Well, first thing is those rate constants vary quite a bit from one well to the next, orders of magnitude between wells. Some of that is due to the abundance of the biomarker. Some of it is due to the fact that the concentrations of the contaminants are very high in the source well. And they're actually high enough to inhibit degradation. And so the first order rate constant is not a uniform entity across the entire flow path. The highest the highest rate constants are associated with a well that's close to the source well, but not the source well itself. Next slide. So I turned all of those different rate constants into a time-weighted average along the flow path. So I wanted to try to get an overall rate constant that would be associated with the biomarkers. Next slide. All right, this is for cis-DCE. That vertical axis is the concentration of cis-DCE in the well in micrograms per liter expressed as scientific notation. And what you'll notice is at the projected rate constant based on that really whopping fast value for K, I mean, it goes far below our ability to measure the concentration really quickly. So the solid line is the projections and the dotted line is the actual measured data. The other thing to notice is that 2 wells really tell the story and the rest of them are really not important. The attenuation is accounted for in the projections from the abundance of markers in monitoring well 21 and monitoring well 30. I just show this to you to give you some sense of the effect of variation among and between wells on this overall average rate constant. Next slide. Okay, so for 2013, we've got 2 field scale rate constants, 5 per year for cis and 6.7 per year for vinyl chloride. And depending on whether we use parameters from the Victoria strain or the Ithaca strain, 195, we get rate constants that are quite a bit different, in most cases quite a bit higher. Next slide. If we just use the DNA markers and then do the calculations for cis, we're overestimating the rate constant by a factor of 5 to an order of magnitude, which is why those projection concentrations are so much lower. But it's off quite a bit, factor of 5 to an order of magnitude. Next slide. And if we did the calculations just using the Ithaca strain, we really underestimate the vinyl chloride rate constants. So what I'm going to do from this point forward is use the Victoria rate constants to try to describe this particular site. Next slide. Because the Victoria cultures are in better agreement with the field data. And at least the ratios between the rate constants for cis-DCE and vinyl chloride are approximately the same. But we do have this issue that we're overestimating the rate constant. Now, in terms of the second line of evidence, what it can say then is that, well, This gene biomarker says there are bugs out there that can plausibly explain what is seen at field scale. It's plausible that they're an explanation. Next slide. I decided to see if it mattered whether I did the calculations using the excuse me, the biomarker for the ribosome or the biomarker for the gene. And so I ran the calculations based on the abundance of DHC and VCRA. Next slide. And it doesn't. Those, there's no real difference between those. And so for this particular site, you could have used the ribosome marker just as effectively to predict rate constants as the biomarker for the particular enzyme that we're interested in ascribing the rate to. Next slide. Well, okay, it's a plausible explanation. There's enough DNA out there to make enough bugs to cause enough degradation to explain what we see at field scale. But a lot of people are bothered by the fact that it's off by quite a bit. So the genes are good, but we'd like to do better. And recently, another alternative biomarkers come into the marketplace. Next slide. This paper is now in its early release in ES&T engineering. The work was done by the Mandy Michelson's group and Kate Kosharik's group, looking at actually quantifying the protein in the enzyme TCEA and VCRA in water from this site. And so next slide. What I'm going to give you is a very cursory overview of what was done with this. This particular topic would deserve an MI seminar all of itself. But basically what was done is they took a standard bioaugmentation culture, cis-DCE9, did metagenomic sequencing to identify the reductase enzymes that are in that particular culture. And then they made some C13 mass labeled surrogates so that they could develop a mass spectrometry approach to actually measure the abundance of peptides that are involved in these particular RDAases. And so what you get is a direct chemical measurement of the quantity of those particular enzymes in the water sample. I stole this basically from a presentation that Mandy and Kate did on validating and evaluating this approach to determine its linearity and its robustness, and particularly its detection limits, to show that this particular approach can see these proteins at concentrations that are low enough that they're relevant for environmental systems. The short answer is yes, they can, and yes, they are. Next slide. Aptum, Paul Hatziker and his group did a microcosm study where they took sediment and they added the bioaugmentation culture, and then they measured rate constants for degradation of cis-DCE and vinyl chloride, and compared the rate constants to the abundance of these proteins to see if the abundance of the protein predicted the rate constant. And the short answer for this microcosm study is they actually did a pretty good job over several orders of magnitude. And so in the laboratory, in a microcosm system, the abundance of the protein predicts the rate constant. And so the question is, then, does this same relationship hold up at field scale? Next slide. All right, so we have some kinetic parameters for the peptides in the literature for the Ithaca culture. And so these are the numbers for Vmax and Km and the reference to Rowe et al. basically I'm going to now use to calculate a field scale rate constant from the abundance of the peptide biomarker. Next slide. So it's the same Michaelis-Menten equation, except now Vmax is normalized to the abundance of the peptides. Next slide. All right, so these are the wells that we sampled earlier this year, and the table gives the abundance of the TCEA and VCRA biomarkers that were measured when we were able to measure it in those water samples. Next slide. And then for these 4 wells, we were able to come up with a calculated rate of degradation of cis-DCE in units per year. And again, monitoring well 21 dominated the story. And then the other 2 wells are not nearly as important. Next slide. And so this is a time-weighted, distant-weighted average of the expected benefit of the rate constant and then the projected actual concentration. And that's a lot closer than we got with the projections based simply on DNA. Next slide. And so this compares the original DNA qPCR data. Then in the bottom slide is the bottom line is the rate constant that's calculated from the abundance of the peptide data for TCEA. Next slide, please. So the proteomic approach got a rate constant that fell within the confidence interval of the field scale rate constant. In other words, this was not different at 80% confidence from the field scale rate. And it's a factor of 2 to 5 or so lower than the rate predicted from just the genes. So based on this one example, that's looking pretty good. Next slide. It is a closer match to the field data than in the constant estimated from the DNA biomarkers. Next slide. All right, so at least we have one field site where we've evaluated and validated the approach. And I showed you a bunch of formulas, and I had several headaches before I got all that figured out, and it took me a long time to do the calculations. And most of you people listening to me today are too busy to do that yourself. So next slide. Anticipating that, we built a calculator, a computer application that does all that for you. And it will be available soon for an updated version of the BioChlor model that was developed under an active ESTCP project. It was supposed to actually have been on the web page, but it didn't quite make it. The model is built, the model is reviewed, the model is submitted to Certif ESTCP. All they have to do is put it up. So it ought to show up on the web page for this particular project very soon. Next slide. So this was done by GSI Environmental in Houston, and they call it the M&A rate constant estimator. And it's designed really to update the old BioChlor model as a screening model for natural attenuation. The original BioChlor was formulated for chlorinated ethenes. It was dated and clumsy to use in modern versions of Excel. The rate constant estimator has a number of new features. It also allows you to look at 1,4-dioxane, and it also encompasses degradation of 1,1-DCE and 1,1,1 trichloroethane, which were chlorinated compounds that's all commonly associated with 1,4-dioxane spills. So it's a little broader in application. It works off a slightly different assumption on the kinetics in the computer model. But one of the tabs models the PCE to TCE to DCE to vinyl chloride to ethene process. Next slide. And so the intended purpose of this is to, by backfitting and matching data using the model to the field data, to try to get a calibrated estimate of the rate constant for degradation with distance along the flow path. And so basically what you do is you turn the knobs on the rate constant until they match the field data. And then you use the rate constants to see how far the plume might go and see if there's a possibility that it would extend and impact a down gradient receptor at concentrations that are unacceptable. So it's really intended for risk evaluation and exposure evaluation. Now it has one little tab that allows you to actually input values for genes that degrade cis-DCE and vinyl chloride to see what kind of rate constant you would expect from the abundance of those biomarkers. Next slide. And so in this particular input, you select the biomarker that you want. I set it up to where it was based on the kinetic parameters for the Ithaca, the Cornell isolate. And it uses the same calculations whether you supply it with VCRA abundances or DHC abundances. You can pick one or the other, but basically it does the same arithmetic. Next slide. And so you tell the model your abundances, your biomarkers, the distribution of the wells, the concentrations of the contaminants for the Michaelis-Menten calculations, and it cranks them and gives you an estimated rate constant, which then you can compare to the fitted rate constant that you got by turning the knobs on the rate constant to make the field data match the model projections. And basically, if your rate constant predicted from the abundance of the biomarkers is close to or above your fitted rate constants, then you've got a plausible line of evidence to explain the rate constant. Next slide. So where does this leave us today? I would submit that the kinetic parameters do allow a quantitative evaluation of the contribution of biological reductive dechlorination. We ought to be getting more information out of our qPCR data than we are. Instead of just simply saying, is anybody home, we ought to ask the question, are there enough of them to matter? Now, I showed you that probably this is going to overestimate their contribution, and that is a reality. But at least you can see if the abundance you have is plausible to explain the rate constants that you need to have to explain your field data. Okay, and plausible is all you need for the EPA second line of evidence. First line of evidence is it really going away? That's what really matters. Next slide. All right, that's the opportunity. There's always a little kicker or a trick in that. These varied widely from well to well, and I really think, based on experience we've had at a few other sites, that's not a unique property to our case study site. Every site has this property that some wells have very high abundances and other wells somewhat lower abundances. And so to make this work, you need more than one or two samples. Obviously, the more wells you can sample, the better. And the more evenly distributed along the flow path, the better. And I also think that at this level of development, you need to sample it more than once. So you need to come back a couple of years apart, depending on how fast the water is moving, and resample this, repeat the sampling and analysis so that you build up enough numbers that you have a central tendency effect on the time-weighted averages. So if you've been sampling your site every once in a while for the qPCR biomarkers, you have that. If you only did it once, this is a good argument, you need to do it again. But I really would not trust this whole approach to just one round of sampling for the reason that I just was very honest and explicit about. The abundances vary widely from one well to the other. Next slide. The other thing that we have is a data deficiency or a hole in the literature. We have that very elegant study out of Ruth Richardson's lab at Cornell for TCEA, where we have the kinetic parameters based on the peptides of the enzyme. But no one's done this similar work for VCRA or BVCA. At least I haven't found it in the literature. So that's an opportunity. We need to do the same thing that was done for TCEA for these other two important reductase genes. And so, you know, these three seem to cover most of the activity against the chlorinated alkenes. So I think it'd probably be realistic coverage once we fill this data gap for VCRA and BVCA. Once we have that, then we can use this proteomic data to get more robust, more targeted, probably more accurate estimates of rate constants. I will say at this point, it's still the second line of evidence. One of the dreams that several of us have had in this field is to be able to measure these biomarkers and robustly predict from them a priori the field scale rate and have it be credible. I don't think we're there yet, but the ability to measure the peptides is a major step forward into meeting that long-term goal. So that's where I see us to be today with this whole approach. What we've got now is useful. What is starting to come online is even more useful. And so I think we need to be answering that second half of the second line of evidence. Not only is anybody home, but are they doing us enough good to count? All right, that should be the talk, Dora, and I'll be happy to entertain questions.
Thank you so much, Dr. Wilson. That was an excellent presentation. We do have quite a few questions that have come in. If you have additional questions, please type them into the GoToWebinar panel and we'll get to as many as possible. So the first question for you, Dr. Wilson, is can you apply this method to sites that were bio-augmented as well as just native populations of the helicocoides?
Certainly. I mean, the bio-augmentation strains are just native strains that have been grown in culture. I can't think any reason why it shouldn't apply. And in fact, the original purpose, one of the original goals from Mandy Michelson's project was to apply it to both bio-augmentation and natural attenuation. So short answer is yes.
Great. Did you guys look at RNA in comparison to the protein work?
Yes, we did. I didn't talk about it. I guess the short answer is because I don't know that I understand it. We did see the RNA biomarkers and occasionally in field systems. But if you think about what the bug is doing with the system, if you're growing a creature in culture and it's in log phase, it is growing, it's making new protein, it's making new cells, it's making RNA. You interrogate that system, you expect to see the RNA. In a natural system, the bacteria often, particularly under M&A circumstances, are not in log phase. They're not growing. They're alive. They've got the protein. They can process the contaminant, but they're not making RNA. And so that tends to, it's not a better, or I think really particularly useful predictor of the behavior of systems that are sort of at the end of the growth cycle, like many natural attenuation systems are. It'd be more useful if you studied something that was growing out rapidly, like a bioaugmentation culture and a active cleanup where you're providing a lot of carbon substrate. I've said more about that than I probably should. We looked at RNA. I think it probably has some importance in understanding active cleanups, but it really didn't tell us what we needed to know about the natural attenuation systems.
How did you find the protein isolation to be from these complex environmental samples? We know that sometimes RNA extractions can be difficult.
I don't think I understand that question. Would you repeat it?
Yeah, I think what they're asking is, RNA, we know that preserving RNA from the field to do extractions can sometimes be difficult, and we've seen changes in lab studies. Did you find that to be the case with the proteomic samples, or was that easier to do?
Well, my comments are informed by some samples that I sent Microbial Insights from another project. And we did see RNA abundances that were reasonably close to the DNA abundances. And so the kit and the processes that were provided us to get those samples and send them back to Microbial Insights did not provide any evidence that we were missing RNA. But we didn't do a formal study of it, and I can't pretend that I'm an expert on the question. All I can say is I tried it and I didn't have any problem with it.
Okay, great. Do you know if there are any molecular tools that can be used to specifically show cis-DCE degradation?
Well, the enzymes that I talked about have a variety of substrates. And so the nature or evolution did not craft these enzymes particularly for cis. And so the tools are designed to recognize the biological entities that process cis-DCE, but they're not specific to cis-DCE. They're wider in their application. The tools to understand whether cis is going to degrade or not is more likely to understand the behavior of the entire biological system. Cis-stall is a process, not a phenomenon. And so it just has to do with competition with other substrates and interactions among enzymes and things like that. So there's not a magic bullet to understand cis. You have to understand the entire system.
Do you know if there are commercial methods that are available for the peptide identification?
I talked to Dora before this presentation, and Microbial Insights has the capacity to reach out and do the TCEA enzyme peptide. And so it's commercialized through Microbial Insights.
Do you know when the updated version of the BioChlor will be available? I know you said that they're putting it on the startup website. Any timing, understanding of that?
I was told that it would be there today. But there's a lot going on in the federal government. I'm sure they're working as fast as they can to get it up, but I can't predict when that would happen. I would hope it's in the next weeks.
Is there a plan to use this in other systems, like in the RIM core?
It's an obvious application, but I don't know of anybody that's working on it at this point. point of it is you could take the M&A rate constant calculator, feed it with the abundances and get a rate constant, and then use that same rate constant in another model. So there's really no need to actually incorporate the package in REMChlor. You just run M&A rate constant estimator, get your rate constant predicted from the Michaelis-Menten equations, and then apply that for any other model that you choose to implement.
That's a great idea. Okay, one of the historical problems with using cell per gene numbers to predict rates is the use of only planktonic cells in the water, not the absorbed cells. Do you have any insight on how we can better do that?
That's been one of the bugaboos of this whole approach for some time, based on some good work that was done by Natalie Capiro and Frank Chappell, and showed that, I'm sorry, Frank Loeffler, that showed that under conditions for good growth, the Halococoides bugs tended to be sticky and stay on the solids. I think that that is a consideration and an issue for active stimulation projects where you provide a lot of substrate. I'm going to talk about a logic that is a little bit circular, and I understand that, but the evolutionary concept is this. If a bacteria has it good, they want to stay where they are. And so the evolutionary pressure is to make them sticky and associate with the solids. If a bacteria is not quite making it, and if they don't find something better, they're going to die, they need to move. And so the evolutionary pressure is for them under those circumstances to be planktonic, because they have a better chance of being dispersed to an environment where in fact conditions are better. Under the M&A circumstances, the bugs have gone through the growth cycle. The concentrations of substrates are limiting. They're starving, they're strangling. And so they have an evolutionary pressure to have the property to disperse. So I don't think the sticky DHC issue is nearly as big an issue with monitored natural attenuation applications as it is for active remediation applications. One of the earlier slides, the one that shows the nice microcosm study that Paul Hatzinger did, there was actually two treatments. One treatment was pure groundwater from one of our hazardous waste sites where we added the bioaugmentation culture. The other had groundwater plus sediment from the site, plus the bioaugmentation culture. And there was no difference between the two treatments. In other words, the presence of the sediment did not change the kinetics, suggesting that under the conditions of the microcosm and presumably under the conditions in the field scale system, the bugs were not sticking to the sediment.
That's great information. The next question is as ISTOC was involved in this research effort, was any of this approach compared to the push-pull direct measurement test he does?
Well, we really want to do that experiment. Jack ISTOC is ready to go. Mandy and Kate are talking to CERTA PSTCP about a next phase where that experiment will be done. And I have high hopes that that will come to fruition.
The next question is interesting. Could this be beta tested using an established rate constant at a site to predict the amount of Dihalococoides proteins that would be found in the water?
I don't understand about beta tested, but it could be reverse engineered.
Mm-hmm.
Yeah, you could run the calculations the other direction and ask the question, how much do I have to get this rate constant? That's really what the M&A rate constant estimator is designed to do. But you could use the formulas backwards. You certainly could.
Okay, the next question says, do you have plans to determine an empirical correlation factor for the Victoria Dihalococoides derived degradation rates by evaluating it at more sites, since it appears high, although accurate, although skewed high, that could help understand the rate constant?
The project that Mandy Michelson led actually did the same thing at 2 other sites. And the other sites were wimpy. We didn't get a lot of activity, so we didn't see enough to actually put another point on the curve. I would say that this process is going to be good for rate constants down to, let's say, 0.1 or 0.3 per year. But at this point, there are not plans to do that. That we're going to have to pass the ball to somebody else. What would be needed is a good field site that had that a rate constant in that range, let's say 0.1 to 0.3, that had enough field data that they could constrain the estimate of the field scale rate constant from the monitoring data tightly enough that you could make it a good comparison. So if you have that field site and you want to do some research, I think you need to make your pitch to CERTA.
Okay, the next question says, this was an excellent talk. Does the kinetic model account for the fact that a concentration well above the Km value, the reaction is 0 order?
Yes, it does. Now, if the value of V is 0 order, now the value for K is V divided by the effective concentration. And so as that effective concentration goes up, then the pseudo first order calculated apparent rate constant goes down. But yes, at our site, we saturated the enzyme system and V was 0 order pretty much.
So the next question asks, if we could use this for pretty much any remedial strategy, whether they're doing enhanced reductive dechlorination or M&A.
Well, that's my hope. I don't see any reason why it couldn't be applied.
Okay, so this one is using a statement that they've heard. It says, as you stated many times, a well is a hole in the ground that tells you lies. The microbes are mostly attached to soil particles, and water from a well is subject to preferential flow paths. I know it's difficult and expensive, but do you know if anyone has done cryolants or soil cores to collect the PCR and proteomic data to see how that compares?
There was a study that Frank Loeffler and Associates did on a site in Michigan more than 10 years ago, one of the first sites to actually validate the process, where they actually recovered core samples. Very few people do that because it's inordinately expensive. The cost of your samples could easily be half the cost of the project. And most people are unwilling to spend that amount of money just to get the samples. So there are very few data sets that have that information or that detail. If the person that asked that question would send an email to you and you forward it to me, I'll be happy to provide that reference. I don't have it off the top of my head. Or they can ask Frank Loeffler, who did the work in the first place, and probably would be the better person to speak to that concern. As far as the other part of it, you need to understand the geostratigraphy of your site before you interpret data from monitoring wells. You want monitoring wells that are screened across the conductive intervals that are going to carry the maximum part of the flow of the system. And so I have a lot more tendency to appreciate and trust a monitoring well that's screened in the sands and gravels. And so that's just basic hydrogeological common sense to do a data quality screening of whether the well is screened in the right place before you start interpreting information provided from the water samples.
Yeah, that's a great point. Do any of these tools or calculations you talked about today take into account back diffusion, like from silty clay?
Not yet. What they provide is an instantaneous rate. In other words, it's based on what's in the water at that point in time. It's not predicting how that rate constant would change over time as back diffusion reestablishes the background concentrations. You could do a sort of a theoretical exercise and do some rim chlor type back diffusion calculations and ask the question, well, what concentration would I have 10 years down the road? And if I had that lower concentration, what would my rate constant be if I had the same abundance of bugs? But as a microbial ecologist, I don't think you could assume that you'd have the same concentration of bugs. I really think the concentration of the active organisms tends to mirror, not necessarily one-to-one correspondence, but follow the concentration of the resource. So I think the back diffusion phenomena is going to change the concentrations, but I think as the concentrations change, the abundance of the active organisms will change. So the short answer is no. But the point to remember is that these calculations apply to today's circumstance. They're not a projection of future behavior.
Can you talk about some of the parameters that should be measured to be able to apply this new biochlor model?
Well, I like to have information on pH. Believe it or not, there are a number of sites where that's out of the range that I'm happy with, or most people that are familiar with the behavior of these organisms is happy. So I'd be careful about applying it to a site where the pH is weird. The reason being that often wells will recruit water from different flow paths. I'd like to have information on, don't get it anymore, but I'd like to have information on hydrogen concentrations. So that's difficult under today's analytical service energy to acquire. But that really is informative about whether there's enough hydrogen to keep these bugs active and happy. The real issue, though, goes back to the comments I made about the geology and where the wells are screened. You need to interpret your well in terms of, is it really in a flow path from contamination up gradient? Because you set up the wells in the model as travel from A to B to C to D to E. And if the water's really not flowing from A to B to C to D to E, then the model is going to give you a nonsense or very inappropriate understanding of the system. So you need to make sure that the wells along the flow path or wells along the flow path. One of the other tools that I often use for that is to look at concentrations of chloride to see if they're different. But that's a limited extent of my bag of tricks as far as that goes.
Okay, one of the questions that came in is interesting, and we still hear this a lot. Can you talk about your opinion of why we should incorporate these molecular tools rather than just simply measuring the chemical concentrations directly?
Well, it goes to the expectations of the Oxford guidance for application of M&A. The regulators under the EPA guidance have a right to expect that you come up with some reason or rationale that shows that you have a right to expect the disappearance of the compound. I talked about flow lines to wells. It is very possible to install monitoring wells that miss a plume. And so if you just look at the concentration data with distance, it looks like the concentration's going away. Well, all you did is you missed it. And I've missed a few plumes in my career. And so the role and the value of the M&A parameters is to allow you to associate this apparent disappearance with the biological process as opposed to just an inadvertent artifact of how the monitoring wells were installed or designed. That's why EPA wants a second line of evidence.
Okay. Another question, there's two last ones that we'll answer since we're almost out of time. Have you conducted any CSIA work on carbon or chlorine that was used to complement this study?
I haven't, but I've read a number of very good papers by the people that do this. And then when people do them both, often the results are very internally consistent. And so the CSA actually is just a second line of evidence. And if you get 2 second lines of evidence that are in good agreement, then you're in a really good situation to say that you understand that, yes, it is disappearing and it's not just an artifact of sampling.
Okay, last question for today. So can you talk about other factors that come to mind that would cause variability between wells other than just the concentration of contaminant, availability of organisms, and preferential pathways? What else should we be thinking about?
I think there's a tendency to underpurge wells. People are trying to avoid the cost and expense of dealing with the purge water. There are many wells where you don't have to purge a lot to get a good sample, but I think there are others where that is an issue. And well, let's see, I'm going to go ahead and say this. We've got a real challenge today to get FedEx to get samples in 24 hours. Our shipping companies have become much less reliable. And so I'm afraid that Microbial Insights and their competitors are seeing samples that are arriving hot. And Dora, if you want to talk to that issue, you can. But it's easy to spend a lot of time and effort to mobilize a sampling crew, get a bunch of samples, send them off, and they don't get to the analyst in time or in a condition that they're not compromised by warming up. So if you want to finish this by talking about whether my supposition is a reality in the marketplace or if it's something that we need to think about, I'm going to pass the baton to you to talk about that.
Well, thank you so much, John. And you're right, shipping has changed over the last couple of years. So we sent out an email a few months ago. If you're interested, email us at info@microbe.com. We'll send you a little document that we put together, but basically pack it with as much ice as you can. And if these are samples that are very important to you, which most of them are, be sure to choose the most priority shipping that you can do, because that'll get you further up the chain and getting those delivered to us quickly. So it is definitely a problem, and we're working with carriers trying to figure out better solutions for it. But right now, the best thing to do is to control as much on your side as you can. And make sure that you get them to us with as much ice as possible, just so, especially when it's summer temperatures, or if they sit in a warehouse for an extra day or outside somewhere for an extra day, that you're not having a problem. Just trying to keep them as cold as you can and choose the best priority on the shipping. But it is a problem. Reach out to us. We'll be happy to send you kind of a little document that we put together with some guidance on helping, especially with FedEx, to get through these troubling times. But I can't thank you enough, John. Your webinars are always phenomenal, and you provide so much value to the industry. It's just an honor to know you and to get to do these with you. So I really appreciate you doing this for us today.
Dora, you're very welcome.
And thank you everyone for taking time out of your schedule. We will have this available on the Microbial Insights website soon. And so you'll be able to listen to this valuable information again. And if you're interested in getting a PDF copy of the presentation, if you can email info@microbe.com, I'll make sure that our customer service department has those available for you to send out. But again, we appreciate your time, and especially your time, John, for doing this for us today. And I hope that you all have a wonderful holiday season and a blessed, wonderful new year. And we look forward to talking to you again on our webinar in January. All right, thank you, John.
Welcome.
All right, bye-bye.